Hello, welcome back to the Computational Genomics Lab. Today we are going to be making the last step for DESEQ is the sample sheet. We made the counts table last time to get the counts all ready for DESEQ input, and now we need to tell DESEQ what samples are what and what statistical analysis to do comparing which samples. Alright, so let's get started here. I'm just going to orient again for a little while here that we're on the Rinn Lab Github. You click on Master Class, and we're here in Lessons, and 05, RNA-seq, and 04, the last file in this folder, last step, and move on to level 6 and start doing some statistical analyses. Alright, so you can follow along here, copy and paste this into an R Markdown file on your RStudio. Here I am in my RStudio, and again we are on 05 RNA-seq in RStudio, and I'm going to click on 04 DESEQ sample input sheet. Okay, so again, the RMD is going to have a lot more detail here, and the videos are going to go a little faster just so you can see and get some concepts, and then go back and read more detail there. Okay, so first let's load that data we saved last time. Remember how handy that was? My environment is currently empty, so I don't have any objects. And 1, 2, 3, we've got that G2S, the gene ID to gene name, counts matrix, and counts filtered. And counts filtered is what we're going to use for the input into DESEQ. But today we're just going to learn some basic R data table manipulation skills again. And so it doesn't matter which one you really use. So let's pull in our raw counts matrix as if we were starting right from here. That is what we did in the last class. Let's see here. Okay, so we just loaded in counts matrix by using read.table, and we have the raw one. So here, this one has not been fixed as we did last time. So let's just go over that again real quick. You can see the gene name is still in this column, and these aren't rounded. But that doesn't really matter for today, because what for today we're going to do is grab these sample names and make a sample sheet of what samples are which. Last time we focused on the rows. Today we're going to focus on the columns and make a sample sheet. Okay, so it doesn't matter which one you use. It's another way of doing this. We're going to make a new object, writing over the name. Again, kind of dangerous, but it's okay for this exercise. Counts matrix is going to be the input into our tidyverse pipe. So we're going to say think about this file, and then select the column named gene name and take it out. That's what this minus sign is doing. And then we can view it. And we see the first column is gone, and we have all integers in here. Okay, cool. Again, that's just kind of a rehash of last time, but what we really want to focus on is those column names. They have the names of each sample, like 0 time point replicate 1, 2, 3, 12-hour time point 1, 2, 3. And I think you'll see why that's a handy way of naming your samples. Okay, so in the DESEQ, we're going to make a thing called DESEQ samples. That's what it's going to need to run. And we're going to use this data.frame function again, very base R, and it's going to run whatever in the parentheses, and simply make one column called sample ID, and we're going to take the call names of count matrix. Now last time we took the row names and use that as to make a column in our G2S. But this time we're going to use the column names and grab them, turn each column name into rows. So we're going to go from this to that. And that's quite common. It's kind of like pivoting in Excel. Okay, so let's start making that. And that call names function is really handy because it'll then grab all the names of the columns. And let's see what we got here, DESEQ samples. Okay, cool. So we have our sample ID. So now we have a unique identifier for every single sample. And what we want to do next is sort of go in there and split this by the underscore, and then we'll have 3 objects, the wild type, the 0, and the 1. The 0 represents a time point, and 1 represents a replicate, and we're going to make a time point and a replicate column using the sample name. Okay, so to do that, we're going to use a thing called string split. You can think of this a little bit like text to columns, where you can do a separator and make new columns in Excel. Except for in R, it's going to be just thinking about those splits and keeping them in a numbered order, 1, 2, 3, because we're going to split on 2 things and get 3 pieces. Okay, so that's the string split function. Again, really handy. We'll use it a lot. And we're going to go into our DESEQ samples, which right now is just this one column. And we're going to go into the column sample ID using the cache sign index. And we're going to use the splitter in string splits function parameters that we have to input. So let's split on the underscore. Okay, so let's do that. And then if we look at it, it's going to be in a different sort of form than we're really used to. So we have the 15 rows, which is each sample's unique ID. And for each row, it's thinking about three things: the first, the second, and the third position after they split. So if we want to grab position 2, we would get the time point for all of these. And if we grab position 3, we would get the replicate number. And that's what we're going to do. Again, lots more written down here. And we're going to use our first function. We're going to do a lot more on functions and make our own functions, in fact. But in this case, we're going to make an object called time values. So we're just going to grab that second value. And to do that, we're going to go into the split values table that we just looked at, which is in this bizarre looking format, but it'll make sense later. And we're going to run a function, x, just a generic function. And we want to retrieve the second value of that function. And so this is where this is one line that is pretty succinct to do a lot of things. So sapply is just like a for loop. So it's going to go through each row and apply the function. The function is x, grab number 2. That's all it's going to do here. So for every row, or it's like a for loop, for I through row numbers, take the split value table, run a function on it, grab value. The function is just grabbing value 2, and x is set exclusively to 2 with a double bracket. We'll see those more, but less frequently than the single bracket indexing. This is just saying absolutely has to be value 2. Okay, let's do that. Awesome. So now we'll see that this is a different type of data. It didn't go into our environment with one of these handy drop downs, and it's just remembering a string of values. And we can take that string, turn it this way, and we'll get the rows, and they're matched identical to the sample ID. Okay, so we're going to do the same thing with replicate values, for loop into split values, run the function, grab the third value, and call it replicate values. That's why the arrow's going that way. Okay, did that, and now we can see the replicate values. So we have time point 1, replicate 0, time point 2, 0 time point, replicate 1, replicate 2, replicate 3, 12, 1, 2, 3. And that's what DESEQ needs, is to say what is the time point if I'm going to compare across time, and what is the replicate number, so I can get the variation within the replicates of the same samples. So this is really handy. So we're going to take our DESEQ samples sheet here, which is just the sample name right now, and we're going to pipe into it, or just use the arrow to put in values. And you can do this, and in fact, this column doesn't exist, time point, but by indexing this way into the object you're making, you're actually going to create that column. So it's going to create a column called time point and pipe in time values. So now let's look at our DESEQ. We have our sample ID and the time point, and this is what DESEQ is going to really take a look at here. Okay, so let's do the same thing for replicate values. Now we should have a third column, and we can see they all match up. This is 0, 1, 0, 2, 0, 3, and so on. Okay, good, we're moving along here. Now one of the last things we're going to do is factor. Now this is very meta, and we're actually just going to say the time point and replicate columns are the thing you're thinking about. So factoring means they have an importance level, if you will. And we're going to factor the time point column here just by using the function factor. Inside the parentheses, we have our DESEQ samples column name time point, which already exists now. And same thing for replicate. And so if we look at our DESEQ samples currently, we'll just see that we have these three columns: sample ID, replicate, or time point, and replicate. And after we run this, we're going to see that change to having a factor there. It just popped up. So it's saying, okay, now I know that this is a group. So I know a group of time points, and I know a group of replicates. Awesome. That's the main thing we need. And so let's save that using our save function. But again, always check, set go to working directory, I'm in the right place, good, okay. Or set as working directory. This is the results folder we made, and we're going to do save our DESEQ sample sheet into and our counts matrix. We don't really need that because we saved it before, but as you wish. So that is it. You now have the counts table all organized and a sample sheet that looks like this, where we're going to put this into DESEQ so it has a unique identifier that knows what time point is represented and which replicate is represented, and it'll use those to do its statistical calculations. All right, so that's it for level 5. Let's move on to level 6 and start finding genes that are regulated by docs across time. All right, we'll see you then.